Hello and welcome to our virtual presentation on our research of building an extremophile to live on Mars.
Here's what we'll be covering today.
Hi, my name is Samyukta Iyer.
Hi, my name is Britney Segovia.
Hi, my name is Lucyk.
Hi, my name is Sklar.
Hi, my name is Haena Vuylsteker.
Extremophiles are defined as an organism that thrives in extreme environments. We wanted to design an organism using already existing traits among extremophiles that would thrive on Mars. We researched a specific area we believed would be suitable for life, selecting the site named Nili-Fosse. Clay and rocks with amounts of silica are found here, caused by a past water presence, and telescopes have detected methane in this area. We also decided our organism we wanted to utilize these traits for would be a potato due to its ability to be genetically modified.
The primary goal was to be able to genetically modify a potato with the traits of existing extremophiles so that it would be able to survive on Mars and make it more habitable for human life. The following list are some steps we took to meet our goal.
In order to achieve our goals, we had to form a plan in which I will briefly explain the methods we chose to follow. We wanted our organism to be located on Mars, which would help us determine which traits to use. After forming this outline, we began researching to gather more information on our area and 5 chosen traits to figure out which organism would provide the best fit. Lastly, we compiled and shared our findings.
We wanted to make our potato photosynthetic to be able to make its own food. The extremophile I chose to look into was cyanobacteria. We plan to depend on the polypeptide protein complex known as PS1 and inject the enzyme ALC because they will both aid in creating energy for the prokaryotic cell in our potato.
My organism was a psychrophile, which are organisms that live in very cold temperatures. They are characterized by lipid cell membranes that are chemically resistant to the stiffening that traditionally occurs when organisms are exposed to the extreme cold. We selected an obligate psychrophile for our organism because they can live in more extreme temperatures.
Since Nili-Fosse's environment has a significant amount of methane, we chose the trait of methanotrophism to be able to assimilate the carbon within methane and generate ATP. We chose the extremophile Methylomicrobium burrietans, which is a type 1 methanotroph that uses the ribulose monophosphate pathway to create this advantageous backup metabolism source.
For the radioresistant trait, I chose the extremophile D. radiodurans for its ability to repair DNA damage. The organism contains a REC-A protein, which helps speed up the recovery of radiation damage done to DNA strands.
For our last trait, we felt that it was important for our organism to survive with little available water. Xerophiles accumulate internal solutes to maintain a high enough osmotic pressure for survival and growth in really hot and dry areas. One specific xerophile is Aspergillus acraceus, which contains the critical enzyme DG18. Being able to add all these proteins and enzymes into our potato will help it survive in areas that lack water, such as Nili-Fosse.
Our results were the correlation between the environment of Mars and the traits given to an organism through genetic modification.
Our project has 3 main future applications. Firstly, for planetary habitation, our project would enable the sustainable long-term habitation of Mars, and this model could also be applied to other habitable planets. Secondly, for disaster relief, we could help terrestrial regions that are affected by natural disasters like floods or droughts to provide them with a consistent food source as well as a way to revitalize agricultural economies. Finally, for in-flight cultivation, as the length of space missions increases, it's important to have a self-sustainable food source to help the astronauts. In conclusion, the genetic modification of the potato with specific extremophilic traits will optimize it for success and long-term growth on Mars. Overall, our experience with the season trip was extremely positive. Although we faced some difficulties with time management and creating our own schedule, we really learned a lot from the speaker series and were able to apply it effectively to our project.
A special thanks to each member of our team, Ms. Henry, Ms. Miller, Ms. Baggio, TSGC, and NASA for giving us this opportunity.
Here are our individual insights, and here are our sources. Thank you.